Hello. Blooming heck, you guys are going a bit crazy, aren't you? Talking about your A levels and GCSEs and my messages. How are you lot all doing? I've genuinely been really enjoying watching this. I had this like screen up for the last, I don't know, few minutes, and I was just watching people saying like some really interesting stuff. But yeah, you guys all right? Anyone actually here, or are we just talking about triple? It's fine. Can you guys see me? There we go. Yes, how am I? I'm really good, thank you. I had a really nice weekend, and I was playing a game called Catan with some of my friends online the other day, which is pretty cool. Various things like that. Good times. Have you guys been up to anything? Has anyone managed to actually like do anything remotely fun? Hello. Just studying. Yeah, I remember those days. Hillary, what are you? What year are you in? Are you year 12, year 13? Or are you just studying because you like studying? Flashcards, very nice. Oh, I love flashcards. You're in year 12. Okay. Yeah, year 12 it's an awkward one, isn't it? Because hey, it's really interesting to like think that you've kind of got to do work now because you know that by the time you go back to school for year 13, you're going to be expected to have done quite a lot of it. So I think year 12, you've actually got one of the most awkward positions to be in, like other than anyone else, because like, I don't know, the school probably can't just restart year 12 kind of where you left off. It's a bit weird. Anyway, yeah, my Easter's been good, thank you. I've had a really nice Easter. I've been working last week. I had like about 4 days off for Easter, which was good. Some of you are year 11s. This is interesting. If you're year 11, I think this can be quite hard. What do I recommend for year 11s? Oh my God. Well, I would make sure that if you didn't finish your course, your GCSE, I'd very much make sure that I was up to date with that and I knew it. So I would try and make sure that I knew all the stuff that I could for year 11. And then I guess if you know what A levels you're doing, you might as well get started looking at that as well. But I would make sure I know all of the year 11 stuff off by heart. Yeah, this is all exam boards. Well, all exam boards. Basically, the main ones that people in the UK actually study are AQA, OCR, EdXLA, and EdXLB. So I've been focusing mostly on those. I know there are some like international ones and there are some other ones which someone always mentions. It's really funny. Each time I do these sessions, there's always one person who's like, oh, what specs are you doing? And then someone says, oh, how long is the session? But I'm doing all of those. The people who do the international ones and stuff like that, basically, there's going to be a lot of overlap. There might be occasional little things where it's like, oh, you might not need to know that. So I don't think any of the boards are particularly harder than others, to be honest. They're all pretty much the same. In fact, they're all very similar apart from EdXLA. EdXLA is a bit of a strange one because they like to make it a bit more practical. So EdXLA is more varied than the other three, but the other three are basically the same. Yeah, CIE. CIE, I will cover lots of stuff from CIE, so don't worry, I'm just not focusing on it. Right, anyway, so let's get cracking, I reckon. So we're going to be looking at some study and cell sort of things. Okay, so well, you'll see what happens. We'll get cracking. Right. So oh yeah, I forgot to mention as well that at the end of this session, as I do every session, I'm going to have a coupon code for you for those of you that are interested in Stamp Revise. So stick around if you want that. If you don't, then don't worry. But firstly, and I haven't got the easy multiple choice questions this time, I'm going straight in with something hard. Can any of you tell me how you would make a temporary mount of a piece of plant tissue to observe the position of starch grains in cells when using an optical microscope? So how do you make these temporary mounts? I'm intrigued to see if you guys know this. And for those of you that have covered wet versus dry, you should be able to work out which one I want based on what we're looking at. So you cut a thin sample. Okay, yeah, that's really good. Thin sample of tissue or something like that. Yeah, use your scalpel to cut a piece of plant tissue. Very good. So I thought this would get some of you a bit confused. You know, when you look at a microscope, right? And you have your little slide and you put something on it. This is called a temporary mount. Okay, so I put this question in because this is like fundamental cell biology, isn't it? Like, how do you make a slide to look at stuff? That's it. That's all it is. Yes, some of you will know this term, some of you won't. Some specs do it, some don't. So I've got my glass slide. I've got my thin section of plants. Someone has said that you need to add some water. Aaron, why would you say potassium iodide out of interest? Good, so you need a stain. Very nice. Macerate the sample of the scalpel and place it onto it. Why are we macerating it? Stain it. Okay. Yeah, no, I was just wondering if Aaron knew this or if he'd Googled it. It looked like he'd Googled it. I don't believe him. But yeah, no, you're right. You've done really well on this. So we want to start out by getting a slide, right? And on the whole, most of the time you need to add a little bit of water to the slide because we're looking at a biological specimen. We don't want it to dry out. So you get your slide and you add a drop of water. Okay, when you've added your little drop of water, you are going to get a thin section. So obtain a thin section of plant tissue. Why does it have to be thin? Does anyone know this? Just need water just to make sure that it's basically not going to dry out. So if you did it without the water, your sample will dry out really quickly. Yeah, good. It has to be thin so that light can pass through it. Remember that a microscope basically works because you've got a light source somewhere down here shining up and your eye is somewhere up here. So essentially it needs to be thin enough that light can go through it. So obtain a thin section of plant tissue, and you might use a scalpel or something like that, and you add it on top of the water. You're quite right that you need to stain it. And iodine would make a very suitable stain here because we're looking at starch grains, and we know that starch causes iodine to turn black. So you stain with iodine, and then you lower a cover slide over. And then if you want to make sure that you always get the mark, and also just before anyone says, oh, is that the same as a cover slip? Yes. To make sure you get a mark, you can say using a mounted needle, which is basically like, it's like a long stick like that. And it's got like a little needle at the end of it. So lower a cover slide gently using a mounted needle. Okay, so I think the purpose of that last sentence is just to make sure that you have got the right piece of apparatus named. So for all of the specifications, they like you to be able to do certain practicals, and every single spec wants you to be able to use a microscope. So this term here is quite an important one, just because it shows that you clearly know how to use a microscope. So you add it gently to prevent any like massive air bubbles or anything like that. Okay. What are we saying? We're saying. Yeah, so it's preventing the bubbles. So temporary mount is just a microscope slide, basically. It's just another way of saying that. One thing which I was going to mention as well, which has temporarily gone from my brain. Oh yeah, someone mentioned that you need to macerate your sample up. So you need to, I don't know, use a needle to start to grind it up and stuff. That would actually be really counterproductive because I'm trying to observe the cell. You only ever do that when you're trying to break up tissues, and we're not doing that. Okay, right. This is a really weird question. I'm intrigued what you make of it. So produce a scientific drawing of a mitochondrion in figure 2 in the box below. Oh, sorry. Right, yeah. Produce a scientific drawing of a mitochondrion in figure 2 in the box below. I haven't got a box. Label the following parts, the matrix and the crista. I know you can't draw this, right? But what are some of the rules that you need to take into account when you do draw a biological sample? It needs to be thin so that light can go through. Thank you, everyone who's replying. So crista are the folds, yes. And so no shading, no sketching, straight lines for labels. Very good. No unbroken lines. Yeah, very good. Sharp pencil, I guessed, yes. Not the most important bit. Add magnification of a title if you've got it, yes. No arrowheads, yes. Good, yeah, very good. So another key thing which you guys probably haven't actually considered is you actually draw what you see. So some people would draw things like DNA in this for some reason, but you're not supposed to. Jack, I don't know why, they're just some rules for sort of the standard rules for drawing a biological sample. So I can see sort of the outer membrane that's coming around a bit like that. Oh, it's like, what's that show on Netflix with the guy? Some of you know what I mean. And then I can see, how many marks? This is 2 marks. So hang on. So I can see that this one sort of, it looks like it's almost floating. So I'm going to sort of draw it kind of floating there. That one's there. That one's coming up there. This one there. It's interesting, yeah, because I would expect that these would be all attached, but for some reason it doesn't look like they actually are attached, which is a bit strange. These next ones look like they are. So maybe I'm going to have my inner membrane coming in here. Something like that. My inner membrane. It's like a little circle, something there. I am not very good at drawing, as some of you might have just noticed. There you go. So something like that is roughly what it looks like. Obviously, I would make sure that this little kind of red splodgy bit down here, I would do my best not to do that. I think it's because I'm using a bit of a rubbish pen. But I would then need to label up what I can see. So this bit here is the cristae. And it also wants me to label up the matrix, which is the sort of gap. I would use a ruler for drawing these lines. But otherwise, that's pretty much all you need to do, right? You don't need to make it any more detailed than that. You basically just need to draw what you see. Try not to have lines that cross each other like that. So, I don't know. If I was drawing it wrong, you wouldn't want to have something that looked like this. And you got like all these little crossy bits. You definitely don't shade anything in because you'll lose loads of marks if you shade anything in. Make sure you don't have any like free floating lines, so you can't have a line here which is just not really doing anything. Anyway, yeah, I think this is quite important to realize this, right? Because you can get asked to do questions like this and most people wouldn't expect it. Most people would think that you're never going to get asked to draw something. Just make sure you know your rules. What happens if you use a laptop for an exam? That is a very good question. I don't actually know. That'd be really intriguing now, I don't know the answer to that. It doesn't have to be exactly, yeah, it has to be what they've shown you. You can't just draw what you think it looks like, you're drawing what they've asked you to. So produce a scientific drawing of the mitochondrion in figure 2. If you joined up the membrane to form a whole mitochondrion. See, I wouldn't because that's not what it looks like. I mean, feasibly, it looks like it might be attached there. That looks like it could be connected up. But this one, that one's definitely not connected up to anything. So it could feasibly be that from this angle, you just can't see where it's connected up. Like maybe if you were looking behind the mitochondria, if you changed the orientation, you'd be able to see where it joined up. But otherwise, I wouldn't. No worries, guys. What's the difference between cristae and the intermembrane space? So the intermembrane space is basically the gap between like here and here. So it's like this little area here. So actually, that's probably not the best way of describing it. And if I draw you what I would normally draw a mitochondria to look like. There you go. So this is the outer membrane coming out here, and this is the inner membrane. So it's going to be like the space in between the inner membrane and the outer membrane. So the space between here and here. That's the inner membrane space. So yes, in terms of other things we could do, I could add a title, which is mitochondrion. I don't have a magnification, so I can't do that. So yeah, that's about it. That's all I can do. I've got 2 things that I can label. I've got a title of it. That's about it. Don't need to be in pen. I would use a pencil. Would I recommend biology over chemistry A level? It depends what you want to do. If you like chemistry, then no. If you like biology, then yes. Right, okay. So a student prepared a stained squash of cells from a tip of an onion root and observed it using an optical microscope. During the preparation of the slide, he cut the first 5 millimeters of the tip of an onion root and placed it on a glass slide. He covered the tip with a drop of stain solution and a cover slip. He warmed the glass slide. He pressed down firmly on the cover slip He identified and counted the nuclei in different stages of the cell cycle. Explain why the student, one, used only the, I don't know why I got it twice. So explain firstly why the student used the first 5 millimeters from the tip of an onion root. So for 1 mark, why is this student only using the 5 millimeter tip? Good. So yeah, why is he only using the 5 millimeter tip? What else are you saying? Good, where meristems are found. Good, so essentially he is doing an experiment to observe mitosis, right? So you won't give a mark if you just say this is the meristem cells. You need to say this is where mitosis is occurring. Or this is where cell division is occurring. Okay, so why did he press down firmly on the cover slip? So cells aren't overlapping. Somewhat that is important. So cells aren't overlapping, I agree. But it's not the best thing you could say. Not to get rid of air bubbles. That's what everyone always says. It's not to remove the air bubbles. Why do you want it to be 1 or 2 cells thick? Like layers thick. So light can pass through. That is the main reason, right? Why is he squishing it down? So part of the reason is he doesn't want there to be like more than 1 cell on top of the other because it makes it really difficult to see. So it should spread them out a bit, but it's also importantly allowing light to move through it. So if I was answering this question, I would say to obtain a sample which is 1 cell thick, so that light can pass through. Okay, so I don't know why they don't like to give this mark for air bubbles, because you are right that this will get rid of some of the air bubbles, but that's not the main reason for doing it. Okay, so just be careful with talking about air bubbles because they are important to get rid of, but it's not as important in making it thin enough to allow the light to get through. So my mark scheme says single dash thin layer of cells spread out, so in bold, light passes through, making the nuclei in cells visible. Okay, so be really, really careful here. You want to be talking about the reason for doing this as to allow light to get through. Do you have a mark scheme with you, or are you just using your own brain? A bit of both. So I do have a mark scheme, and I'm using it every now and then to make sure I'm saying the right thing. I can, yeah. So single dash thin layer of cells dash spread out so that light passes through, allowing cells to be visible. Something along those lines, right? So you need to get the word light in this answer to get the mark. Okay, right. Here is a nice little mathsy question, just to make sure that our brains are all functioning and doing what they should be doing. So figure 1 shows the cells the student saw in one field of view. He used this field of view to calculate the length of time these onion cells spent in anaphase of mitosis. Scientists have found the mean length of time spent by onion cells in anaphase of mitosis is 105 minutes. Let's get a highlighter out. Okay, so it's 105 minutes that they normally spend in anaphase. They also found that the cell cycle of this onion root, shown in figure 1, is 1080 minutes. 32 whole cells are shown in figure 1. Use the information in figure 1 to calculate the length of time the cells of this onion root are in anaphase. And then calculate the percentage difference between your answer and the mean length of time found by the scientists. Show your working. Right. Have a go at doing this and see what sort of things you think. And yes, Aaron, if you are being irritating, then stop it. Otherwise, we'll just get you removed. Yeah, this question is mental as well. Oh, there you go. Aaron has been timed out for 300 seconds. And don't worry, I'm sure he probably doesn't know what he's talking about as much as he claims he does. How many marks for this? This is a 3-mark question, which is going to make you feel really bad about yourself because it's so long. So, okay, scientists have found that the mean length of time spent by onion cells in anaphase is normally 105 minutes. So most onion cells are 105 minutes, or spend 105 minutes in anaphase. Okay, they also know that the cell cycle is 1080 minutes for onion cells. Using information in figure 1 to calculate the length of time that the cells of this root are in anaphase. So how could I work out the amount of time that the cells in this diagram are in anaphase for? How could I work that out? Feasibly. Right, how many cells here? How many cells can you see in anaphase, firstly? Yeah, so, whoops. So that one is, and that one is. So 3 of them are in anaphase. How many are there in total here, does it say? It says 32, doesn't it? So 3 out of 32 are in anaphase. And then if I'm working out how many minutes are therefore spent in anaphase, what do I need to do? This is something quite important. This is a really easy calculation, which lots of people don't know. You don't multiply it by 105. Good guess, though. Nope. Yeah, you multiply by the total time. So what you do is you do 3 over 32 multiplied by 1080, because that's the total cell. So what we're doing here is we're saying that 3 out of 32 of these cells are in anaphase, so the rest of them aren't. And therefore, if we times that by the total cell cycle, that shows us that at this snap point of time, I know that 3 out of 32 times the overall time, that's how many cells are in anaphase. So 3 over 32 times 1080 equals 101.25. Okay, so in this picture, that is the amount of time that I can see that they are spending in anaphase. So that's in minutes. Okay. Now the scientists found this actually 105. So they found that the mean length of time is 105. Okay. So you've been asked to calculate the percentage difference between your answer and the mean length of time found by the scientists. How do you do that? So lots of you are getting to a good answer. Nope, that's not how you do a percentage difference. Percentage difference is honestly one of the most important equations you need to be able to do. Right, it comes up in every exam I've ever seen. Percentage difference is, yeah, the difference between the two divided by the original. So 105 minus 101.25. Which equals whatever that equals. I'm not even going to bother doing it because I don't need to. And then you divide that by the original number that they gave you, which in this case was 105. That was what they found the amount of time spent in anaphase to be. So 105 minus 101.25 divided by 105. So if you do that and you times it by 100 because it's a percentage, you get an answer of 3.57. So that is a percentage decrease. So you could write 3.57 or you could write 3.6. That's also fine. Strangely, with this question, they actually allow you to use the other number. So if you did 105 minus 101.25, if you instead did it over 101.25, they'll give you the mark if you do that as well. Right? So either way around that you do it, they'll give you it because there's not really an original number. They're not asking you to say if it's an increase or a decrease. They're just asking you for a difference. So you can do either or here and it's fine. I would just stick with 105 just because that's the original number that they gave me. Okay, so the answer is 3.6 and that's for 3 marks. Okay, that is pretty much all you need to do for that question. I know that's quite a hard one. But anyway, I thought you'd appreciate looking at that. Is it acceptable to use 105 and 101.25? Yeah, you can use either or. In many papers, yeah, so they do allow it in lots of papers. Just go with whatever the original one is on the whole. Or if they say a percentage decrease, then you know that you're going to go from, you can use 105. If you're using a percentage increase, then you need to use 101.25. Okay, cool. Next then. So statement resolution that can be achieved, and I only want a rough one for this. Statement resolution that can be achieved by each of the following types of microscope. So what is the rough resolution of a light microscope? I definitely want some units in here. Definitely slightly different in what you guys are saying here. So I would expect that a light microscope will be somewhere about, and it's normally measured in micrometers here. So a light microscope has a resolution in micrometers of about 0.1 micrometers. Okay, normally about that. Okay, an electron microscope isn't too much harder to remember because it's about 0.1 nanometers or thereabouts, right? So I will accept a range of answers here, and I've actually got it in, so anywhere from 0.1 up to 1, 2. Sorry, I'm just dividing something in my head. So 0.1 to about 0.2, I'll accept here. So that's actually not a very big gap. And this one will be about 0.1 up to about 1 nanometer, which is a huge gap. Okay, so for this question, I think this was an edXL question, they will accept anywhere between these values, right? And it changes because obviously newer electron microscopes are going to be slightly better, older ones are going to be slightly worse, but on the whole, that's roughly what you should be aiming to remember, okay? Yeah, I would be happy with either of those. What if you gave a light microscope in nanometers? Yeah, you can do that. It would just be 1,000 times smaller, wouldn't it? No, wouldn't it be 1,000 times bigger? So you'd need to do it as not 0.1, but 100. Okay, we're going to get to SEM-TEM in a second. The use of a microscopy has greatly enhanced our knowledge of cell structure. Out of interest, what is the difference between magnification and resolution? I will get to the EMs, don't you worry. Yeah, AQA needs to be roughly aware of resolution differences. So what does magnification mean? What does resolution mean? So resolution is clarity. Yeah, I'd like a little bit more than that though. So it's kind of how clear it is. Magnification is how much bigger it looks. Minimum distance between two objects appearing separate. That's very good. Ability to distinguish 2 points as being separate. Yeah, very, yeah, right. So lots of you know what resolution is. So resolution is the ability to distinguish 2 points as separate. Or it's the distance between those 2 points that you can tell that are separate, right? So for instance, you can tell very clearly that those 2 points are separate. So the resolution right now is definitely bigger than the gap between those 2 points. But if I made it absolutely tiny, down to a certain point, you're not going to be able to tell that difference as two separate objects. So resolution is the level of detail or the ability to distinguish 2 points as separate, or the distance between 2 points distinguished as separate. This is all based on what you're using. So it's either the wavelength of light or the wavelength of an electron. So either of those are fine. Magnification, I think lots of you said this, is sort of how many times larger an object appears to be. So how many times larger an image is than the object or than the specimen. Right, you could really define this by an equation. So you could really define this by basically saying the magnification is the image size divided by the actual size, right? It's the difference between how many times bigger the image is than the actual size. Yeah, Kelsey, that's fine. So, but yes, Teresa, equally good. Okay, So figure 4.1 is an electron micrograph showing part of the nucleus. So that's really cool, in my mind anyway. So a student stated that figure 4.1 was taken using a scanning electron microscope. What evidence supports this student's claim? Does it either mean, yeah. Yeah, these are nuclear pores, these little holes. So yes, why does 3D, why is the 3D-ness, why is that significant? So there's something else. So visible nuclear pores, it's the outside structure. Yeah, so Massimi, you actually get a mark for that. So you can either say that it's 3D or that it's showing the external structure as opposed to internal. So yeah, it is really cool. You can actually, we've managed to take photographs of DNA before. If any of you are interested, feel free to Google electron micrograph of DNA. You can just about see it. You can't see it properly coiled up. You can kind of see a shadow of what it is. Yeah, so this 3D thing, right? It's important that you understand why. So it's because the electrons, essentially, with SEM, what you have is you have a little sample down here, and you have something which can make electrons up here. So the electrons are going to come down. And instead of having like a really thin sample that the electrons need to go through, they just scatter off it in multiple directions. So the electrons will scatter off in loads of different directions. There'll be some sort of means up here of detecting it. And then a computer will put that together and tell us what it actually looks like. So the reason that I know it's scanning is because it's 3D, because that's what a scanning electron micrograph allows us to do, or a microscope allows us to do. And it's also showing us the external structure. So a scanning electron microscope cannot show you the internal structure unless you cut it open. Okay. The difference with TEM is that TEM has the electrons going straight through. So a TEM would give a 2D structure, and it's normally showing you the internal structures. It also has a much greater resolution as well. Can you get a mark for saying surface instead of external? Yeah. No, that's fine. If you said surface, that's fine. Yeah, so yeah, no worries with that. So TEM would show you what's sort of in between the nucleus. Yeah, kind of. Would you get a mark for saying black and white? No, because black and white only tells you that it's electrons, right? So anything with a light microscope is coloured, but either electron microscopes will show black and white. So no, you can't get it for black and white, definitely not. Cool, yeah, that's pretty good. Okay, on figure 4.1, the nuclear pore complex labelled A is 3 millimeters wide. Calculate the actual diameter of the pore in nanometers. No, electron microscopes do not show colour. You have to use something called false colour. So basically a scientist, if they want to make it colourful for a textbook, they'll just colour it in whatever colour they want it to be. That's why you get all these funky pictures of like viruses looking like crazy colours. It's not true. We don't know what they look like in terms of their colour. Okay, so yes, it's not 3 millimeters wide in real life. You're right to spot that. They're talking about the image. Okay, so first step, if I'm using nanometers, I probably want to change this into nanometers because I don't like it being messy. I'd probably rather change it all at the beginning, right? So 3 millimeters equals 3,000 micrometers. And some of my students used to get a bit more, GCSE students in A-level, used to get confused at how you know whether to make it bigger or smaller. But we all intrinsically know that a micrometer is very, very diddy, right? We know there's not a lot to it. So 3 millimeters is going to contain absolutely loads of micrometers. So just logically you get in there, you should be able to work out if it's bigger. So nanometers is 3 million. Okay, now I'm going to use the equation IAM, which is one of the few that you ever need to really learn in A-level biology, at GCSE as well. And then I need to work out basically, have they given me the image size or the actual size? Well, someone said earlier that this cannot be the actual size of it, so it's got to be the image. So I'm doing 3 million over 25,000, which is 120? Everyone get them up? Yes, good. Ooh. So pretty small, right? Like it's not getting too far off of the absolute minimum that you can get to. Like I can tell those two things as being separate, right? So clearly the resolution is lower than 120, otherwise I wouldn't be able to see that. But yeah, it's a weird structure, isn't it? It does really look like the surface of the moon. It's very strange. Okay, so 120 nanometers is my size. Awesome. Yeah, sorry, the 25,000 is coming from here because I know the magnification, right? I know the image size, so I'm doing I over M equals A. Okay, here is a horrible question, right? I thought I'd have one absolute belter of a horror. So, apologies. A hemocytometer is a special microscope slide which can be used to count the numbers of blood cells in a sample of blood. The surface of the slide has a small square, has small equal-sized squares marked on it. The depth of liquid in each square is 0.1 millimeters. Right, let's get the highlighter out again. When counting cells that touch when counting, cells that touch the top or the left lines are counted, but cells that touch the right or bottom lines are not. A doctor used a hemocytometer to determine if the number of red blood cells per millimeter cubed in a blood sample. He diluted the original sample by a factor of 200 times before putting some on a hemocytometer. The diagram shows the distribution of cells in a typical small square. Okay, the doctor counted the red blood cells in many small squares. The mean number is 7. So the mean number is 7. So that's really key. The original blood sample was diluted by 200 times, as we already know. Calculate the number of red blood cells per millimeter cubed in the original blood sample. Right, so this is key as well. You need to know that it's they want it in millimeters cubed. So your units are going to really help. Give your answer in standard form. A doctor probably would never do this. Yeah, I imagine they have better things to do. Okay. Right, so how many cells did they say were here? Oops. 9? Don't think so. Yeah. It's literally, they've said 7, right? They've said the average is 7. Doesn't matter. I don't actually have to read anything or count any of this, right? They did say somewhere that you count the ones that touch the right. Sorry. Cells that touch top or left are counted. So 1, 2, 3, 4, 5, 6. It is 7 anyway. So 7 cells. What is the volume here? What volume have they got here? How do I work out the volume? That's an area, Valencia. Yes. So to work out the volume, right, because I'm ultimately trying to work it out in red blood cells per millimeter cubed. But at the moment, I know that this little square is like a square like that. That's 0.5. That's 0.5. But they actually said that the depth is 0.1. So it's very tiny. It's like a little cube. Okay, so this line here is 0.1. Okay, so to work out what my volume is, I need to do 0.5 times 0.5 times 0.1, which gives me 0.025. Okay, so there are 7 cells in 0.025. Is it millimeters? Yeah, millimeters cubed. Okay. Now, I need to work out how many red blood cells there are per millimeter cubed. So if I want to turn this into 1 millimeter cubed, what do I have to times this number by to turn it into 1? In fact, I've done something wrong, haven't I? 0.05 times 0.05 times 0.1. I was way off there. Sorry, apologies. So that's 0. yeah, low, way off. So 0.00025. There you go. 1, 2, 3, 4. 1, 2, 3, 4. There you go. We good? That was me being stupid. So 0.05, because I wrote 0.5 down, I think, which is why I went wrong, times 0.05 times 0.1 is 0.000025. Yes, good. So in order to turn this number into 1, right, because I'm timesing this by some random number, right, and I don't know what that is. So the easiest way to do this is, instead of timesing it, I'm just going to divide it. So I'll do 1 divided by 0.000025, and that will give me this random number here, right? So if you do that, 1 divided by 0.00025 is 4,000. So Jacka, I made a mistake. So if you do it on your calculator, 0.05 times 0.05 times 0.1 is not 0.25. That was me being very, very stupid, which I very occasionally do. So times 1,000 is 1 millimeter. So what do I need to do to this number then? My blue screen's in my way. Oh, right, okay, hang on. I've got a solution to that. If I start writing down here, can you see down here? Is that all right? Can you see that? Cool. Right, so I've times this by 4,000. So on the other side, I need to times it by 4,000. Whatever I do to one side in maths, I have to do to the other side. So times 4,000. Okay, so in 1 millimeter cubed, I would have 7 times 4,000, which is 28,000 red blood cells. Is there anything else that we need to take into account with this question? What is that? Yes, lovely. Well done, Asio. So last point then is he diluted it by 200 times. So the last thing I need to do is times that by 200. Which will give you a big old number. Okay, so I'll go with this one more time, but it gives me the number of 5.6 times 10 to the 1, 2, 3, 4, 5, 6. Okay, so going back over it very, very quickly then, for those of you who didn't understand that, and I'll get rid of this top bit because my face is in the way, as it always is. So I firstly worked out that there were 7 cells, because it told me that there were 7 cells. I then was clever enough to work out that this volume is 0.05 times 0.05 times 0.1, which equals that. Okay, they wanted it, so the unit they gave me was per millimeter. So I needed to see if I've got 7 in that tiny amount, then how many would I have in 1 millimeter? Well, to do that, I need to know how many times bigger 1 millimeter is than 0.000025, which happened to be 4,000. So I multiplied 7 by that to give me 28,000. And then it did say up here, he diluted the original sample by a factor of 200. So if I've diluted it by 200, then at the end, I just need to multiply it by 200, and that will give me the right number. How is that worth only 2 marks? I know. This is one of those questions where if I saw this, unless I was very confident that I'm good at maths, I would probably leave until the end and then just come back to the end and give it a go, because why not? Okay, right. How many more do I have? I've got like a few more questions. So I might not do that one because it's too long. And this one's a good one. This one's a good classic one, which is really standard. And then I'll do one final one. Okay, so a biologist prepared a sample of organelles from, a biologist prepared a sample of organelles labeled C from the liver. He used the following method. So he added the liver tissues to ice cold buffered solution with the same water potential as the liver tissue. He mixed the liver and the solution in a blender. He then filtered the mixture from the blender. He then spun the filtered liquid in a centrifuge at a low speed. He made something called a pellet in the bottom of the centrifuge. He poured off the liquid from that pellet into a second centrifuge tube and spun it at a higher speed to obtain the organelles able to see. Basically, this is something that AQA like to talk about a lot. So the other specs are sort of less into this, but I'll go for it anyway. So explain why the solution the biologist used was ice cold, buffered, and the same water potential as the liver tissue. So why would scientists want to make sure that it was ice cold? Asio, you are totally right. So the reason that scientists like things to be cold is so that enzymes stop working efficiently. Okay, so it will slow down enzyme activity. So think about it. If you've got like a big old cell, right, then one of the organelles that you guys know about is called a lysosome, which contains loads of hydrolytic enzyme called lysozyme. If you split open this membrane and that membrane, then all of this little enzyme will start like mixing about and it will start to degrade the cell. So you have it really cold so that the enzyme is working really inefficiently, basically. It's buffered, good, to maintain the pH. Okay, so a buffer is something that makes sure that the pH stays the same. Why does he have the same water potential? Yep, you were right with pH. So we could say that if you want. So it does say explain. So maintains pH, enzymes not denatured or something. In fact, we could say not just enzymes. We could say, no, why not? Enzymes not denatured. It accounts for all proteins as well. You don't want it to be the wrong pH, otherwise lots of things start to break. Good. The same water potential means no osmosis. Preventing osmotic lysis. Lovely. So that's going to stop the cells from being damaged, essentially. Osmotic lysis, if you haven't come across it, is where you have a cell and if water moves into it, if it's an animal cell, because there's no wall, it will basically just burst. Okay? Okay. Right. I'm going to go back to my previous little slide in a second if this doesn't make sense. But finally, describe how you could use cell fractionation to isolate chloroplasts from leaf tissue. Close my window because someone's doing work and they definitely shouldn't be. Yes, sorry, Sean, you're right. But that's going to prevent osmotic lysis. No, Synfugia, that is all you need to know. It's cell fractionation for OCR. It's not so much OCR, no, this is actually more AQA. So this is basically the question we just had, right? So I don't know why I put it here, but fractionation is when scientists don't want to study what it looks like, they want to study what's actually there. So if you wanted to study what's in the nucleus, for instance, this is what you do. So essentially, you want to have your medium, which is going to be cold. So you need a solution which is ice cold, buffered, and isotonic. Okay, you then need to basically open up your cells. So you need to homogenize, which basically means blend in fancy science words. Homogenize your tissue. And you will almost certainly need to filter it as well. Okay, and then the final bit is ultracentrifugation. So ultracentrifugation is what you've probably seen in like labs on the TV where they have something that spins around really fast. So ultracentrifugation is carried out. Ultra just means it's fast. And essentially what happens is when you spin it once, you'll have all of the heavy stuff, all the dense stuff moves to the bottom and it forms a little white solid, which is called a pellet. And all the stuff which is less dense basically forms a liquid, which is called a supernatant, which is a great word. And essentially, the heaviest organelles will be in the first pellet. So the first time you spin it, you'll get mitochondria in the first pellet. So ultracentrifugation is carried out. The pellet will be removed, right? Pellet containing nuclei is removed. However, the supernatant will then be spun again. So supernatant spun again. And you need to make sure that you do it faster. It's respun faster. And this forms chloroplasts and mitochondria in the pellet. Bit of a hard question, that one. But that's one that most people don't actually revise very well. In the pellet. Lovely. There you go. That is fractionation in depth. That's pretty much all you can need to know. When we need to separate the mitochondria from the chloroplast, yeah, that would be quite difficult. I don't actually know how you do that. There's probably like a fine line where one of them is slightly more dense. So feasibly, the chloroplasts are a little bit more dense than the mitochondria. So feasibly, if you could spin it for a certain amount of time, you get the chloroplast out first. Okay, so is this only AQA? Yeah, this is AQA. What else did I say? Yeah, it's quite nice. There are some things where certain people don't need to know certain things. But anyway, guys, I'm going to finish here and I'm going to very quickly tell you about why Snap Provides is the best. And I'm trying to think of fun ways of doing this each time, right? So fun way of doing this today. So imagine that you had a really, really long day and you were like, oh, God, everything's going bad. Got to cook my own dinner. And so doesn't know how to, I don't know, be my friend anymore, and I'm really quite sad. And I know I've got all that revision to do, and all that revision's rubbish, and I'm just feeling really down about everything. Well, I have a solution for you. I'm really sorry, this is so cheesy. The solution is Kadoosh, Snap Provides. If any of you are remotely interested in this, basically, I do these lessons literally all of the time. So if I show you the dashboard of Snap Provides, check this out. Let's say that you do AQA biology or OCR biology. Why not? Right? Our website not only has a web classes, so I'll show you web classes first. Essentially, this is what I do every day of my life. So I'm not doing any tomorrow for reasons, but on Wednesday, I've got web classes. Thursday, I've got web classes. Monday, web classes. Tuesday, got 2 on Tuesday, 3 on Tuesday, Wednesday, and then I need to add one for Thursday as well, which I haven't added yet. So I do web classes. I do drop-in tutor sessions where I'm around to answer questions. Look, I do a biology book club as well. Think of all the fun you could have. So not only do I do these web classes, and that's like the very top end of our website, but we also have really good means to revise generally. So let's say you don't know very much about mitosis. All right, let's say you're rubbish at mitosis. No one's commented from my side. So basically what our website does is it'll get you to do a quick little quiz, which will take about 1 to 3 minutes. So I'm going to get it all wrong on purpose by clicking here. Always B. Oh, why is it never B? It's always C, isn't it? No, it's not. I've always found that C has been always the correct answer most of the time when in doubt. There you go. So it tells me that I got 1 out of 7 correct. I might apparently know metaphase randomly, but I don't know chromosomes. I don't know the mitotic stages. I don't know prophase, anaphase, blah, 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 blah. What our website does is it will show you a whole load of videos, and this one's a bit of a small topic, so not that many, but it will show me some videos where it'll basically go through it, right? As soon as I've done that, it will then give me a little quiz that I can have a go at doing to check that I've understood it. So here's a second quiz, and hopefully I'm going to do better on that one than the first one. It'll give me an exam question that my man will go through with you. Here he is going through a question, and he'll tell you what the answers are. And then after you've done that, if you're still confused, there is an exam pack for you to go through. So you can start going through these questions, some of which I wrote myself, and you have solutions to all of your problems. So let's have a look at this one, a longer answer one. There you go. We have that. We also have revision guides as well, so that you can essentially download all of our stuff and keep forever and not distribute to your friends. I don't know why it's not loading up, probably being slow. But essentially, we've got all of this really good stuff. So if you are finding that you are in year 12 or you're in year 13 and you're trying to do a bit more work and you're struggling with how you can best do it, I don't know, our website's really, really genuinely good. And we've got like, we cover pretty much everything you could ever need to know. Like we have mock papers as well and everything. So if you are enjoying this sort of style of teaching that I'm doing, then please do sign up. If you feel like you just need videos to explain, I don't know, what excretion is, then again, just like check it out, right? I'm pretty sure you can get a free account that gives you some access for like a few, I don't know, for a little while, right? So you can definitely get some access to some of this stuff. I want to show you one other thing. I can't find a video. Basically, if someone writes a question here, this question comes straight to me as well. So I get all of these questions that pop up and I just answer them. So quite a lot of my day is spent just answering people's questions. So again, if that is something that totally interests you, then you should definitely sign up and I'll show you a way to get a better discount in a second. If not, if you are not convinced, which I understand some of you may not be convinced, then keep coming to our lessons. Basically, why not, right? You've got probably nothing better to do at the moment. I am doing another, well, I'm doing one right this second apparently, but I'm doing another one fairly soon. There you go. There's one on the 22nd, so in like 2 days. But there's also maths, chemistry, physics, and they're happening basically all the time. So if you're not convinced, then at the very least, watch some YouTube videos and get convinced. But otherwise, I think I'm probably going to end this here. So those of you that have been around for this whole time, thank you very much for coming. It's been an absolute pleasure. But otherwise, I think I am going to head off. In fact, no, I'm not. I'm going to show you this first. There you go. There is the discount. Ta-da! How come there was only one bio home study last week? Oh, because I didn't do one on Monday because it was a bank holiday. I'm not working on a bank holiday. So yes, next session is on Wednesday. Awesome. Is there a yearly package? Ooh, I don't know if there's a yearly package. I think you can pay by month, but you can feasibly get, you can feasibly pay for a whole year if you wanted to. Nice. I'm glad it's really helpful, guys. If you have any questions, feel free to, I'll stick around for a bit. Bye, everyone else. So it depends what package you go for based on how much it is a month. So I can't remember how much the cheapest one is, but I think the most expensive one's like £50 a month, which does feel like a lot. And I understand that, but you would get how many lessons? I think it's about 48 lessons per month from me. So yeah, it works out being like the most ridiculously cheap tutor you've ever had. So I used to be a tutor, I used to teach, I used to be a teacher. And then before that, when I was at university and I had no money, I used to charge people about £40 an hour for my time to go over like A-level biology and like GCSE maths and stuff. So it's very, very cheap compared to that. It's 24 a year for the base pack. Okay, J Summers, if that is what it is, then yes. Awesome. Right, guys, I'm going to head off then. Have a lovely afternoon, and I'll catch you on Wednesday. But yeah, go to chemistry and go to physics and go to maths. Why not? Even if you don't do them, just go. Where do you add the discount code? I think there's a place to add it. And if you can't find it, just email support@SnapRevise and they'll tell you. Should year 11 start looking at A-level content? Hang on. Can we do these on Zoom so it's more interactive like a lesson? Fatima, I don't actually know. I think it'd be too difficult with a number of you here. So feasibly not, but I can always run it by my boss. Should year 11 be looking at A-level content? Harrison, if you know all of GCSE off by heart and you are getting like A stars in biology and chemistry and physics or whatever you want to do, then yes, look at A-level, but make sure you learn your GCSE stuff first. Awesome. Right, have a lovely afternoon, guys, and I'll catch you later. Any other ways that they would say it, like reword and mean the same thing? I don't think so, no. No, year 11, you should do A-level biology. It's really good. It's a really good subject. If you like biology, definitely do it. All right, anyway, see you later, guys.